So our last speaker before the break is John Perrier, who's a graduate student in Ed Preisdale's lab here at the CVR. He'll be speaking about coagulation initiated by tissue factor on a coronavirus. John.
Thank you, Hugh. Today I'll be speaking about a molecular basis for the hemostatic irregularities that are seen during envelope virus infection, specifically focusing on human coronavirus. I'm just going to grab the pointer here. So to start us off, the main players in this are tissue factor, which is a transmembrane protein that acts as a cofactor for clotting protease factor 7A. When 7A binds to tissue factor, it enhances the activity of 7A to cleave factor 10 and ultimately produce 10A. 10A can leave this complex and contribute to the downstream generation of thrombin and eventually lead to a fibrin clot formation. Now, tissue factor is not only important in blood clotting, it's also important in cell signaling and inflammation, and it does so, one of the ways that it does so is through protease-activated receptor activation, and this can occur through the tissue factor 7A, 10A complex or through that thrombin. Now, my lab has shown that tissue factor can exist on the surface of envelope viruses and that it can actually participate in this cell signaling through either that thrombin generation or through that tissue factor 7A, 10A signaling. And the virus that this is best characterized in is herpes simplex virus 1. So this is an envelope virus which derives its lipid bilayer from the host cell that it's produced in. And we can use electron microscopy where a monoclonal antibody that's conjugated to a gold bead is used to label tissue factor. So we can see here a herpes simplex virus particle that's labeled for tissue factor. And this is important because in vivo, mice that are infected with herpes simplex virus 1 that contains tissue factor have productive infection of the virus. So we can see in plaques per milligram here, there's lots of viral infection when tissue factor is present. However, if you take away tissue factor, there's no infection. So it's required for infection for HSV1. So we hope to expand these results to other viruses that dysregulate the hemostasis system, such as coronaviruses. And we're using human coronavirus 229E. So this is a common cold coronavirus. We can see here on this image on the left that under electron microscopy, we can stain for the corona that's formed by the spike proteins which surround the virus particle. And under electron microscopy immunogold, so again, a gold bead conjugated to a monoclonal antibody, we can see single labeling of virus particles for tissue factor. On the right here, we've also done immunoblot analysis for the cells that this HCOV229E is produced in, so HUH7 cells, as well as the purified virus preparation itself. So they both have the expected molecular weight band present for tissue factor. So following identifying antigen, the next step was to look at whether or not tissue factor activity was seen here. And to do this, we did a 10A's assay where we add purified virus as the source of tissue factor, along with purified factor 7A and factor 10 in calcium. We allowed these to incubate and form factor 10A, which can cleave a chromogenic substrate and produce a color change. So on this graph here, we can see on the x-axis, which is in log, that as we add more and more virus particles, there's an increase in 10A being produced up until we reach a plateau. The next step was to show that this 10A generation by the virus was dependent on tissue factor. So we employed a monoclonal antibody specific to the 7A binding site of tissue factor, as well as NAPC2, a tissue factor 10A specific inhibitor. And in both cases, this abolished 10A generation for anti-tissue factor as well as NAPC2. Interestingly, the virus also contains anionic phospholipid that's sufficient for the binding of these coagulation factors. So we used lactadherin, which competitively binds to anionic phospholipid. And when lactadherin is present, 10A is also reduced. The next step was to see whether or not, so the virus can produce procoagulant 10A species. We wanted to see whether or not it can overcome intrinsic anticoagulants in plasma. So for this assay, we used purified virus as well as pooled human plasma. We initiated the reaction with calcium, and then we measured the amount of time that it took for a fibrin clot to form. So we can see on this axis here, as we add more and more virus particles, there was a dose-dependent reduction in clotting time. So HCOV229E can trigger a plasma clot. And again, using monoclonal anti-tissue factor as well as NAPC2, these are both able to prevent that clot from forming. So no clot forms past the cutoff of our assay of 700 seconds. So to summarize today, I've shown you that we can identify tissue factor on the virus particle using immunogold electron microscopy, that this virus is also capable of generating procoagulant 10A as well as thrombin that we saw through our plasma clotting assay, and that this virus also contains anionic phospholipid that we've seen through the lactoferrin binding experiments. So this is important because HCOV-210E can infect tissue factor-bearing cells, such as macrophages and monocytes, and that this could provide a basis for other thromboinflammatory pathologies that we see in coronaviruses, such as SARS-CoV-2 and MERS. And this is also building towards the idea we've observed tissue factor in oral herpes, dengue, HIV, and now a member of the coronavirus family. So this is the idea that any envelope virus that's replicated in cells that have tissue factor can express it on its surface, and that it's important for both coagulation and infection. And I'd like to take any questions.
I give the question back to you because Ed asked my student regarding the PSI tissue factor interaction. I asked you whether this virus could, through the surface tissue factor interaction with integrin, or use the integrin as a receptor for virus entering the cell.
Yeah, that's a great question. So you're just to confirm, you're asking if beta-1 integrin can participate with tissue factor and have an effect on infection for this virus?
My question is how many copies of tissue factor on this virus surface, and whether this tissue factor can interaction with the cell surface integrin, particularly in PSI domain. Because we did find PSI domain binding to tissue factor. So viruses do have tissue factor on the surface. My question is if tissue factor copy number is sufficient on the virus surface, why this virus cannot use the tissue factor, touch it to the cell surface integrin, then come into the cell?
That's a great question. In terms of copy number of tissue factor, I have completed Western blot analysis that's semi-quantitative to try to determine how much tissue factor there are per virus particles. And that varies for each virus. So for 229E that's produced in the HH7 cells, I don't know if it would be sufficient for binding with integrin. But we do also hope to look at whether or not there are those tissue factor modulators like integrin present on the cells, and if the virus itself can express integrin. So yeah, it's something that I should look into. So for HCOV-210E, it's about 70 molecules of tissue factor, I believe. Yeah.
Very nice work. Thank you very much.
Nice talk, thanks. Do you think that, are you thinking that there is sufficient tissue factor and sufficient viremia in some conditions that it could really be prothrombotic, that it could account for thrombosis in a patient with viral infection?
Yeah, that's a great question. So for this virus, for HCOV-210E, we don't have, there's not good literature on it becoming viremic. So this virus specifically is more to try to, have a molecular basis to understand if other coronaviruses can do so. But for other coronaviruses such as SARS-CoV-2, we also don't know if like you said, the viremia is high enough for it to explain that.
So you think maybe it has another function, like for improving the chances of the virus getting into a cell, for example?
Yeah. So like we saw with herpes simplex virus 1, that tissue factor can be responsible for modulating those protease activated receptors. So I think that it could be expressed on the virus for that reason, to make the cell more permissible to infection.
Yeah, so we published a paper regarding the coronavirus, that's SARS-CoV-2. We found that RGD PEP motif in the RBD receptor binding domain, sometimes it's confusion. RGD sequence in the RBD domain that can interaction with alpha-2b beta-3 integrin, binding to the platelet, trigger platelet activation. I would think that probably initiate that vaccine-induced thrombotic thrombocytopenia in the initial phase. So that subsequently could drive like HIT, like response, immune response. My question here, if we put all this stuff together, whether RBD the interaction with the integrin and also tissue factor could also bonding to that, that's made not only that not affinity issue, but could increase the local affinity, local multiple bonding interaction with SARS-CoV-2. That's probably is additional process for this virus into the host.
Thank you.